Hello friends, I hope you all are doing good and watching my videos regularly and finding it informative and understandable. So this video is about the major receptor families in pharmacology and their drug signaling mechanisms, that how the receptor actually react when they bind to a specific drug, and what are the mechanisms that are involved in their downstream signals procedures. So let's see that what topics we are going to cover in this video. Number one is the receptor ligand interaction, that is all about the chemistry of the receptor and ligands, that how the ligand attaches to the receptor and how the receptor react when it binds to the ligand, what kind of conformation changes, what kind of bonds it forms. Second one is the signaling mechanism, that is downstream signaling procedures of the receptor, that when it binds to a ligand. This is very important in pharmacology because it's all about the mechanism. So the receptor families. So while discussing the receptor families, we should keep in mind that each family of receptor in pharmacology has its own specific mechanism of signaling. So this is very important to discuss about the receptor families. So before going to the receptor families and their mechanisms, we should know the terminologies receptor and ligands. So receptor, as it is a self-explanatory word, it is any biological molecule that upon binding with drug molecule undergoes some conformational changes and produces a pharmacological effect. So each receptor has a specific mechanism, has a specific downstream signaling mechanism, so keep in mind. Next one is the ligand. Ligand is any drug molecule. It may be a enzyme or it may be a hormone that when bind to a specific receptor, it produces a specific pharmacological effect. So I use the word specific for both the receptor and ligand. It means that both of these are very specific in nature, that bind to produces a specific pharmacological effect. Let's move to the next slide, which is about the interaction of the receptor and ligand. So basically, when we discuss the interaction, the chemical bond comes to our mind. That means that when receptor bind to a ligand, there is always a formation of chemical bond. That chemical bond may be electrostatic or hydrogen bond, as well as it may involve some weak van der Waals forces. So interaction basically determines the specificity of the receptor, which means that the interaction, or we can say that the strength of the bond is inversely proportional to the distance present between the receptor and ligand. So in order to make sure the bond, it is very important for the ligand to exactly fit into its complementary receptor atoms. Most of the bonds are reversible, except some of the drugs that bind to their receptors through covalent bond, and these are irreversible. So the size, shape and charge distribution of the drug molecules basically determines which of the binding site in the cells and tissue of the patient that's going to interact with the ligand. So let's discuss the metaphor of the interaction of receptor with its ligand. And this is the lock and key model. This is very simple to understand, as we know that a specific key is going to unlock the specific lock. In the similar way, a specific ligand gonna bind with a specific receptor and it will unlock the activity of that receptor. Before unlocking the activity of the receptor undergoes a conformation changes, and that conformation changes showing that the receptors are flexible in nature. So the properties of the receptor we're going to discuss in the next slide. But before discussing that, some of the summary of this whole slide is that interaction always involves the chemical bond. Secondly, the interaction determines the specificity. Third, the size, charge distribution and shape of the drug molecule determine that where they're going to bind in the cell or in the tissue. And The last but one is um, uh, these interaction is very specific. So let's move to the next slide, which defining some properties of receptors. So most of the receptors are protein in nature. They are flexible and they are very specific. They are non-rigid. They are non-rigid and flexible properties, showing that they undergo some conformational changes uh, when combined with a ligand. So let's discuss some signaling mechanism and receptor families in pharmacology. So here we have some signaling mechanism, lipid soluble ligands that crosses the cell membrane easily, and they interact with those receptors which are present intracellularly. Next one is the receptor that are linked to ligand gated ion channels, and this is very important. Third one is the receptor that are linked to G proteins, and these receptors are also known as GPCRs. Next one is a receptor whose intracellular enzymatic activity depends on binding of the ligand to their extracellular domain, which means that these receptors have two domains, intracellular and extracellular. Extracellular domain going to bind with the ligand, while the intracellular domain of the receptor will precede the enzymatic activity. The last one is a receptor that upon binding to the ligand activate the intracellular tyrosine kinase. That is another protein. So let's discuss the first one, which is the lipid soluble ligands. So most of the biological ligands are lipid soluble, which means that they can easily cross the cell membrane and act on the intracellular receptor. Different ligands are available which are lipid soluble, and these includes the hormones like thyroid hormone, steroid, mineralocorticoids, corticosteroids, vitamin D, and sex steroids. So let's discuss that how they actually act on the intracellular receptor. Basically, they attaches to specific part of the DNA, we can say that, which is the response element, and binding of the ligand to the response element regulated the transcription of the gene active. Gene active is a part of the DNA which is going to be regulated, or which is like we can say that the gene of interest is basically the gene active which we want to be transcribed. So let's discuss the mechanism of glucocorticoid actions briefly. uh, the when there is no hormone available, the receptor binds to the heat shock protein 90, which is responsible for the prevention of normal folding of many structural domains of the receptor. As soon as the hormone is available and it binds to the ligand binding domain of the receptor, it releases the heat shock protein 19, which exposes the DNA binding domain and transcription activating domain. So this lead to altered transcription of specific genes, and this lead to downstream mechanism. So the two important therapeutic consequences of such interactions are all such molecules produce their effect after a lag period of 30 minutes to several hours, and that is due to the time required for the transcription process. So synthesis of the new protein always takes time, so this lag period is basically due to the transcription factor. Next one, the effect always produces by the lipid soluble ligands remain even for several hours to days, even after the level of the drug reaches to zero, and this is because of the slow turnover of several enzymes and proteins. So this was all about the ligand soluble in lipid. So the next one is ion channels. Ion channels are a self-explanatory word, which means the channel which involve the movement of ions across the cell membrane. So these channels produces their effect, or we can say that they transmit their signal across the cell membrane by increasing the transmembrane conductance of relevant ion, and in this way they alter the electrical potential across the membrane. Most of the naturally present ligands are acetylcholine, glutamate, GABA, serotonin, All of these are synaptic transmitters. So let's see an example of ion channel. The most common and most easy example of ion channel is nicotinic acetylcholine receptor, which are present in the cell membrane of most of the neurons and at the postsynaptic neuromuscular junction. So this receptor is basically a pentamer which is made up of 4 different polypeptides, 1 alpha chain. This is a pentamer which is made up of 4 polypeptide subunits, 2 alpha chains, 1 beta, 1 gamma and 1 sigma. The molecular weight of this receptor ranging from 43,000 to 50,000. As soon as a receptor binds to acetylcholine, it opens the central aqueous channel for the sodium, and it causes sodium influx in the cell. As a result of that, the electrical depolarization of the cell take place. So the time elapsed between the binding of the receptor with the ligand and production of the signal is measured in milliseconds, because this depends on the conductance of, or we can say that the movement of ions from movement to movement. So this is why the action produced by ion channels is very quick. It is the most quick action produced. So voltage gated ion channel is another example of ion channel. So the major difference between the voltage gated ion channel and the ion channels, ligand gated ion channel, is that the voltage gated ion channels do not bind to the neurotransmitter directly. In fact, they are controlled by the membrane potentials. Such channels are also very important drug targets. Drugs that regulate the voltage-gated channels typically bind to the site of the receptor different from the charged amino acid, and this is basically the voltage sensor domain of the protein. And this is used for channel opening by membrane potential. So this was all about the ion channels. So which receptor or which channel, or we can say we can ask that which signaling mechanism is the most quick, is the answer is ion channels. The third receptor family is ligand regulated transmembrane enzymes. So as a name, transmembrane enzymes, which means that they have some enzyme which is present which is present in the transmembrane region. So some of the ligands which are producing their pharmacological effect through this signaling mechanisms are some growth factors, which are the platelet growth, derived growth factors, ectodermal growth factors. Second one is insulin, which is an hormone. Next one is the anti-natriuretic peptide, transforming growth factor beta, and some other trophic hormones are the ligand for these receptors. And the enzymes which are transmembrane enzymes which are involved in this kind of receptors are tyrosine kinase, serine kinase, and guanylyl cyclase. So this depends. So let's see the structure of the receptor. The receptor basically involves two domains: extracellular domain and intracellular domain. Extracellular domain, which is a hormone binding domain, and it binds to the hormone or any ligand we have discussed earlier. And the intracellular domain, which is a cytoplasmic domain, it is bind to the enzyme. So it is an enzyme linked domain. The receptor activity begins as soon as the hormone binding domain binds to a ligand or any hormone. So this causes a receptor to undergo some conformational changes, and as a result of that conformational changes, the receptor molecules bind and form dimer. So this dimer formation of the receptor activate the cytoplasmic domain, which contains the tyrosine kinase residues. As soon as the tyrosine kinase residues in the cytoplasmic domain of the receptor activate. This causes phosphorylation of the receptor, as well as the phosphorylation of some downstream molecules, which causes the signaling mechanisms to proceed further. Let's take an example of insulin that uses the single class of tyrosine kinase receptor, and as soon as the insulin binds to the extracellular domain of the tyrosine kinase receptor, it causes the two receptor molecules to dimerize and form a dimer, and as a result of this, the intracellular cytoplasmic domain activates the tyrosine kinase residues and causes a phosphorylation of the molecule, and this signaling mechanism is responsible for the uptake of glucose and amino acids, and also regulate the metabolism of glycogen and triglycerides in the cell. These receptor has its own significance in cancer treatment, because in some cancers, our basic aim is to inhibit the cell differentiation. So in this way, some of the anti-cancer drugs or the inhibitors of these receptors bind to the receptor and inhibit the binding of growth factors. The intensity and the duration of the action produced by epidermal growth factor and platelet-derived growth factor and other agents that act on these receptors is basically limited by the process called the downregulation of the receptor, because sometimes the excessive binding of the ligand to the receptor causes the receptor to the receptor causes the endocytosis of the receptor, and the de novo synthesis of the receptor is slower as compared to the endocytosis of the receptor, and that endocytosis of the receptor is followed by the degradation of the receptor. So very important point to be noted at this is that most of the cancer that are associated with these type of receptor is actually due to the failure of downregulation process, and this is due to the mutations in the gene. As the receptors are failed to downregulate naturally, the growth factors continue to bind on the receptor and causes the cell to differentiate and increase in number. A different number of regulators of growth and differentiation, including TGF-beta, act on another class of transmembrane receptor enzyme and phosphorylate the serine and threonine residues in the intracellular domain of the receptor. Arterial natriuretic peptide, an important regulator of blood volume and vascular tone, act on transmembrane receptor whose intracellular domain is linked to a guanylyl cyclase enzyme. So different enzymes are involved in this receptor, but the mechanisms is the same. The next class, which is similar to that of the tyrosine kinase receptor, is cytokine receptor. Cytokine receptor respond to heterogeneous group of peptide ligands, which includes not only the growth hormones, in fact several interferons and erythropoietin, growth regulators, and differentiation regulators. So the mechanism is quite similar to that of the tyrosine kinase receptor, but in this the protein tyrosine kinase activity is not actually associated with the intrinsic to the receptor molecule. In fact, the receptor is firstly binded to the another protein, which is a JAK, which is a Janus kinase enzyme, which is another protein. And as soon as the receptor binds to the ligand, the receptor dimerize itself, and then they activate the bounded JAK, which is bound to their intracellular domain. As the JAK activate, it causes the phosphorylation of the tyrosine residues on the receptor. Phosphorylated tyrosine residues on the receptor cytoplasmic surface, then causing the complex signaling dance by binding another set of protein, which is a STAT. STAT stand for actually the signal transducers and activators of transcription. So the bound STATs are themselves phosphorylated by the tag. Two STAT molecules dimerize, and finally the STAT dimer dissociates from the receptor and travels down to the nucleus, where they regulate the transcription of specific genes. So this is about the cytokine receptors. So cytokine receptors and the tyrosine receptors are sort of similar ones, but the major difference is that the cytokine receptors are actually bind to the JAK, and JAK activation causes a downstream signaling mechanism to take place. So the last receptor family is the G protein coupled receptors, which are also known as GPCRS. So many extracellular ligand are actually produces their action by increasing the second messenger in the cytoplasm, and these messengers may be CAMP, that is a cyclic adenosine monophosphate, maybe calciumite, or maybe phosphainositide. So let's discuss one by one all of these second messengers. So let's see that how the receptor actually activates. So whenever the receptor binds to the ligand, it undergoes some conformational changes, which means that the GGDP will be converted into GGTP, and that GTP is an activated form of the receptor, G coupled receptors, and this causes activation of the enzyme. The activation of the enzyme is terminated by the hydrolysis of the GTP into GTP again, and the reaction will be terminated at this. So first discuss that there are different kinds of the G coupled G proteins, GS, GI, and GQ, and each of these G protein couple has a different mechanism. GI is basically for the inhibition of the receptor, and it involves the inhibition of CAMP enzyme. Secondly, GS is a stimulatory enzyme, and it causes the activation of adenylyl cyclase, and so the enzyme which is a factor in this case is the adenylyl cyclase, a protein that converts intracellular ATP into CA. And after being activation, the GS coupled proteins are responsible for the action to be proceeded. Next one is the GQ, that is quite complicated. As soon as the receptor binds to the ligand, the hydrolysis of basically the effector element in GQ is phospholipase C. So simply we will start it with that as soon as a receptor is bind to the ligand, the receptor first converted the GDP form into GTP, and this activates the phospholipase C enzyme. The phospholipase C enzyme results in the hydrolysis of phosphatidylinositol biphosphate into DAG, that is a diacylglycerol, and inositol triphosphate, which causes an increase in the intracellular calcium ion. So in this way, the second messenger would be the calciumite. When the GS is activated, the second messenger is CAMP, right? So this is all about the G protein coupled receptor. Some of the facts that is about this family is it is the largest receptor family. It is a 7 transmembrane family, or we can say that the serpentine receptor, because the polypeptide chain of the receptor forms a snake-like structure across the plasma membrane 7 times. So in this image you can see that when the epinephrine and beta adrenergic, that is the beta adrenergic receptor, when epinephrine bind to it, it is actually bind to the when the beta adrenergic receptor bind to the epinephrine, the receptor converts the GDP into GTP form. So see that the GTP is attached to the alpha subunits, and this GTP then bind to the effector element, which is the adenylyl cyclase, and convert the adenylyl cyclase ATP, and with the help of this adenylyl cyclase enzyme, the ATP is converted into CAMP, which convert the protein kinase A into other proteins. So this was all about the receptor families. I hope you find it understand and informative. So in any if you have any queries and questions regarding this video, you can ask it in comment section. And if you have any other topic in your mind that you want me to discuss in the next video, you can also mention it in the comment section. Thank you.